Good morning, students. Today we'll discuss about the urine formation. So, urine formation involves three main processes, namely glomerular ultrafiltration, reabsorption, and secretion. So, what are the three main functions? One is glomerular ultrafiltration, second one is reabsorption, and third one is secretion. The first step in urine formation is the filtration of blood, which is carried out by glomerulus and is called glomerular filtration. On an average, 1100 to 1200 milliliter of blood is filtered by kidneys per minute, which constitute roughly one-fifth of the blood pumped out by each ventricle of the heart in a minute. So 5000 milliliter in a minute blood is pumping out, the heart is pumping out the blood. So that one-fifth of that blood, that means 1100 to 1200 milliliter of blood filtered by the kidneys per minute. The glomerular capillary blood pressure causes the filtration of blood through three layers. What are the three layers here? Endothelium of glomerular blood vessels and epithelium of Bowman's capsule and a basement membrane between these two layers. You can see that one. What is that? This is Bowman's capsule and this one is Bowman's capsule and this one is afferent arteriole. Three layers are there. Capillaries ki endothelium and Bowman's capsule ki epithelium, and in between the membranes are there. That is called the basement membrane. Okay. So that is called the basement membrane. Three layers are involved here for this filtration. That is also called ultrafiltration. Why? That we'll discuss now. The epithelial cells of Bowman's capsule, called the podocytes, are arranged in an intricate manner so as to leave some minute spaces called the filtration slits or slit pores. So we can see here, this is actually three layers are there. What are the three layers? The endothelium of the capillaries, blood capillaries, and epithelium means outermost membrane of which one? Now Bowman's capsule. Now epithelium of the Bowman's capsule, they are arranged in some intricate way, means some slits, some we can say the spaces, minute spaces are remaining between the epithelium of this Bowman's capsule so that blood can filter so finely through these membranes that almost all the constituents of the plasma except the protein pass out the lumen of the Bowman's capsule, pass on to the lumen of the Bowman's capsule. Therefore, it is also called as process of ultrafiltration. You can see that one, ki afferent arteriole diameter and efferent arteriole diameter. So diameter of afferent arteriole is wider than the efferent arteriole. So the blood amount of blood or volume of blood which is entering in the glomerulus, that amount of blood is not coming out through efferent arteriole, and that's why a pressure is created. Due to that pressure, the bloods are filtered out, and in case of the Bowman's capsule, the epithelial cells are known as the podocyte, and that podocyte cells are we can say the arranged in some integrated in such a way that the slits are very minor spaces are kept in that their epithelial cell, and through that pores the blood is filtered out. So except the protein, all the constituent of plasma filtered out here into the lumen of the Bowman's capsule, and therefore it is considered as a process of ultrafiltration. Okay, so first function, or we can say the first process of urine formation, that is called the glomerular ultrafiltration or glomerular filtration, is now over. Second one we are coming. The amount of the filtrate formed by the kidneys per minute is called the glomerular filtration rate. So what are the filtrate are coming out from the glomerular to the Bowman's capsule, that amount of filtrate formed by the kidneys per minute is called the glomerular filtration rate or GFR. GFR in a healthy individual is approximately 125 milliliter per minute or 180 liters per day. Which one? GFR. The kidneys have built-in mechanism for the regulation of glomerular filtration rate. What is that built-in mechanism? One such efficient mechanism is carried out by juxtaglomerular apparatus or JGA. What is JGA? JGA is a special sensitive region formed by the cellular modification in the distal convoluted tubule and the afferent arteriole at the location of their contact. So you can see that one. Juxtaglomerular apparatus is formed by which one? Ki afferent arteriole and this distal convoluted tubule. The modification of these two cells of the present here when they are coming in contact, that is called JGA. Means distal convoluted tubule is the renal tubule, part of the renal tubule which is present far from the Bowman's capsule. So that distal convoluted tubule ki cells of distal convoluted tubule and we can say the afferent arteriole cells, they are actually modified and when at the point of their junction, when they are jointed or when they are actually contacted. So that is called JGA apparatus or juxtaglomerular apparatus. JGA is a special sensitive region and modification of distal is over. Now afferent arteriole at the location of their contact. A fall in GFR mean glomerular filtration rate, when it is decreased, can activate the JG cell means juxtaglomerular cells to release the renin one actually hormone which can stimulate the glomerular blood flow and thereby the GFR back to the normal. So GFR, how the JGA is working here, what is the mechanism here? Now when any fall in glomerular filtration rate जब ये कम हो जाएगा, can activate the JG cells means juxtaglomerular cells to release the renin which can stimulate the glomerular blood flow and thereby the GFR back to normal. So they are actually stimulate the which one? The glomerular blood flow, the blood which is coming to the glomerulus, and definitely the GFR also increased in that sense. A comparison of the volume of filtrate formed per day. What is the filtrate form per day? 180 liter per day, and with the urine is released is 1.5 liter. So filtrate is formed here 180 liter per day and urine is released 1.5 liter. So therefore we can understand that 99% of the filtrate has to be reabsorbed by the renal tubules. This process is called reabsorption. So GFR is coming, GFR means glomerular filtration rate. So glomerular is filtrating every day 180 liter, but the urine is coming out. What is the urine? Urine is here in case of ureter, the urine is stored and urine is coming out in the day. We can say the 1.5 liter. So out of 180 liter, 1.5 liter is released. So remaining that must be reabsorbed in the renal tubule. The tubular epithelial cell in different segment of nephron perform this either by active or passive mechanism. So for example, substances like glucose, amino acid, sodium plus, etc. in filtrate are reabsorbed actively, whereas the nitrogenous waste are absorbed by passive transport. Okay, reabsorption of water also occurs passively in the initial segment of the nephron. So here it is coming, some are actively absorbed and some are passively absorbed here. So reabsorption is occurring here, where it is occurring now throughout the renal tubule. The reabsorption is occurring throughout the renal tubule, but in different segment, different we can say the components are absorbed. They can be absorbed by actively as well as the passively also. So actively, just like the nutrients means glucose, amino acid, sodium plus, they are actually absorbed which one? Actively, reabsorbed actively, whereas that nitrogenous waste and absorbed by in the passive transport. Nitrogenous waste like urea, uric acid, these are actually ammonia, they are actually absorbed passively. Water also absorbed by passive transport. Okay, now we are coming to the reabsorption of water also occurs passively in the initial segment of the we can say the renal tubule or nephron. During urine formation, the tubular cells secrete substances like H plus, K plus, ammonia into the filtrate. So this is called secretion. This is called reabsorption. Third function is secretion. So H plus, K plus and ammonia into the filtrate. Tubular secretion is also an important step in urine formation as it helps in the maintenance of ionic and base balance of the body fluids. So here three steps already discussed. We have discussed here glomerular filtration. We have discussed here reabsorption, who are reabsorbed, how it is reabsorption is going on, and some also secretion, means tubular secretion also happening, just like the H plus, K plus. You can see here H plus, K plus and NH3 plus, they are actually secreting in the tubular part or in case of filtrate. That is called the tubular secretion. Now function of the tubules. What are the functions here? Now proximal convoluted tubular PCT. PCT is lined by simple cuboidal brush border epithelium which increases the surface area of reabsorption. Nearby all of the essential elements and 70 to 80% of electrolytes and water are reabsorbed by this segment. So electrolytes are also absorbed there. As I say, NaCl is absorbed here, nutrients are absorbed here, and water also reabsorbed by this segment. And PCT also help in maintain the pH and ionic balance. So pH and ionic balance of the body fluids by selective secretion of hydrogen ions, ammonia and potassium. So H plus, K plus and ammonia is secreted and others are absorbed. Water absorbed, electrolyte absorbed, nutrients absorbed. So some are absorbed and some are secreted. Who are secreted? H plus, K plus, NH3. They are secreted in the proximal convoluted tube here, and into the, and again it is coming into the filtrate and by absorption HCO3 from it. So as HCO3 also absorbed here. Look at here, HCO3 is absorbed here and these are secreted. So this is the function of which one? Proximal convoluted tubule or PCT. This portion is called PCT, and then between Henle's loop and then it is called DCT. The proximal distant portion is called DCT here. Henle's loop reabsorption is minimum in its ascending limb. However, this region plays a significant role in maintenance of high osmolarity of medullary interstitial fluid. That will discuss also. So medullary means this one. So high osmolarity of medulla, this is called medulla. So they are, this is the ascending limb. Ascending limb, this one is descending limb of which one? Now Henle's loop. मतलब जो नीचे की तरफ आ रहा है, that is descending one, and the which is coming upward, that is called ascending limb. So ascending limb in case of the Henle's loop, we can say the reabsorption are minimum in its ascending limb. However, this region plays a significant role in maintenance of high osmolarity of medullary, and this one, this in between the space, medullary, we can say the interstitial fluid. The descending limb of loop of Henle is permeable to water. They are permeable to water but almost impermeable to electrolytes, and this concentrates the filtrate as it moves down. The ascending limb is impermeable to water but always transport the electrolyte actively or passively. Therefore, as the concentrated filtrate pass upward, it gets diluted due to passage of electrolytes to the medullary fluid. So when the descending limb is there, they are permeable to water but impermeable to electrolytes. So water is coming out, so that the filtrate will become concentrated. जब water is coming out, definitely filtrated become, जो filtrate मतलब inside the renal tubule, what is Henle's loop, what is present there, so that is becoming concentrated. Then when it is coming upward, means when in the ascending loop, that is impermeable to water but electrolytes are again can allowed actively or passively. So you can see that NaCl is coming out, and that's why the concentrated filtrate when pass upward, it gets diluted due to the passage of electrolyte to the medullary fluid, because when these electrolytes are coming where? In the medullary fluid here. So we can see that one here impermeable, this one is impermeable to water, so water cannot come out but electrolyte can come out there. So the filtrate again become diluted. Here it becomes concentrated and again it becomes diluted. Then distal convoluted tubule, conditional reabsorption of sodium plus and water takes place in this segment. So Na plus and we can say the H2O, they are actually absorption is there. DCT is also capable of reabsorption of HCO3. You can see here also HCO3 absorbed and PCT also HCO3 absorbed. And selective secretion of which one? Hydrogen plus and K plus, hydrogen plus, K plus and NH3 plus again, and to maintain the pH of the sodium potassium balance in the blood. Now it is coming about the collecting duct. The long duct extend from the cortex of the kidney to the inner parts of the medulla. Large amount of water could be reabsorbed from this region to produce the concentrated urine. This segment allows the passage of small amounts of urea into medullary interstitium to keep up the osmolarity. Means little bit amount of urea, small amount of urea is coming where? Now medullary interstitial space. So here this one is medullary interstitial space. So when the exchange is occurring between this renal tubule or collecting duct or here, so osmolarity is maintained there. It also plays a role in maintenance of pH and ionic balance. So when the exchange is occurring between the medullary interstitial space and the renal tubule, so osmolarity as well as the pH as well as the, we can say, the ionic balance of the blood, that also maintained, as well as it is also maintained by the selective secretion of H plus and K plus ions in case of the collecting duct also. Secretion of which one? H plus and K plus ion. So this is total function. The different parts of the renal tubule are secreting and absorbing different, actually we can say the nutrients as well different components. So nutrients are absorbed, NaCl electrolytes are absorbed here, water also 70% absorbed here, and HCO3 also absorbed here. But who are secreting? H plus, NH3 plus, K plus is secreting here, and we can say the into the filtrate, and absorption of HCO3 is happening here. And next one we can say this is the secretion is actually maintaining the pH and ionic balance of the body fluid. And next one in Henle's loop, the descending loop is actually permeable to water but impermeable to electrolytes, so it become concentrate. And then when it is coming up, they are impermeable to water and permeable to electrolytes, so that is happening again, the filtrate become diluted again. And this exchange of the electrolyte that makes the with the medullary interstitial space, that is actually making the which one? pH as well as the osmolarity is maintained there. And then in the DCT part, what is happening again? H plus, K plus and H3 is secreted here to maintain the pH and we can say the osmolarity and ionic balance of the body fluid, we can say the blood, by the selective secretion H plus, K plus and H3. Here HCO3 also absorbed, here Na plus absorbed, here H2 absorbed. And then collecting duct also, there also urea is coming out in the medullary interstitial space, so that the again osmolarity and pH will be maintained there. And then it is coming about the here also secretion of which one? H plus, K plus is also there. So these collecting duct are pouring their secretion where? In the medullary pyramid, and then the calyces, we can say the pelvis portion, already have discussed earlier. So here the collecting ducts are coming from cortex to medulla region and pouring their secretion here, and then we can say the urine. So in this process, urine formation is now over, and mechanism of concentration of the filtrate next we will discuss.